Thank you for inviting me for this talk. We're going to talk a bit about some biology, a bit about genetics, and a bit about methylation and the integration, and especially about federated analysis. So how do you do that? So at a larger scale. So slightly different view as the talks before, more from a kind of epidemiological view rather than from a computer science view. So since the last 15 years, there are many epidemiological studies conducted to identify genetic risk factors for disease risk. And it has led to more than 70,000 genetic associations to common diseases and traits, for example, type 2 diabetes, blood cell counts, BMI, etc., etc. However, this is like a massive advance over the years. 90% of those genetic variants are located in non-coding regions. So they're not located in a particular gene, but outside the gene. And therefore, we don't know what they're doing. So although we find an association, we don't know what kind of mechanism is behind that. And the idea is that that the other parts of the genome, which has like regulatory elements and transcription machinery and other kind of regulatory machinery, is contributing to that. So if you have someone has conducted such a genome association study, then that is not the end, because the next thing is then trying to figure out what the association has been doing. And to be able to do that, we can use other types of data, including epigenetic data. I will talk about that a bit more. Transcription data, so that's on your RNA levels, protein levels. So we go, basically what we do, we identify those genetic variants that associate with an outcome, kind of disease risk. And then we think that like somewhere in the middle, there's an intermediate trait. Some of those, we call them omics layers, which could be anything regulatory, and that then will lead to the disease. So there are a lot of different interpretations of the term epigenetics, but one of the generally agreed definitions is the study of mitotically heritable changes in gene expression that occur without changes in your DNA sequence. So if you put that in another way, so basically epigenetic studies how genes are switched on and off by a mechanism that persists to generations of cells. But they are not, so they're different from your genetic factors. One clear example is every single cell in your body has the same DNA sequence, whether it's a skin cell or a blood cell. However, they have completely different functions. So there must be something else on top of that that is regulating that. So, sorry, clicking. So there are several epigenetic mechanisms that fulfill the definition of epigenetics. This include DNA modifications, where chemical groups change the way DNA is read by attaching to the DNA itself. So you have your DNA strand, and their metal groups are added to your DNA strand. And those metal groups are attached to cytosines, one of the 4 bases in your DNA. And then we say, something is methylated. So overall in the genome, you have like 28 million of those CPGs in a human genome. And those, they tend to be like clustered in so-called CPG sites. And then often there are like at the start site of your gene. So you have your gene here, and then before that, you have CPG islands that regulate whether your genes are switched on or switched off. So when DNA is methylated in one of those regions before, in the promoter region of the gene, it usually that blocks the transcription of the gene, so it becomes silenced, and your genes will be switched off. And when DNA methylation occurs outside of that promoter region, so in another region, for example here, then that might have another role. But for now, we're going to focus on those two scenarios. So you have something methylated, your gene gets switched off. It's unmethylated, your gene gets switched on. So you have all those DNA modifications. So your DNA methylation sites, those 28 million, they are kind of variable across the genome, and if you compare them across different individuals, then you can identify factors that influence them, whether they can be methylated or unmethylated. The most obvious one is smoking. Is your an alcohol that will change the methylation status. Another one is that they are cell type specific. So, for example, in blood you have different cell types, and for example, in your neutrophil it can be methylated, and then in your red blood cell it's unmethylated. Other things like genetics play also an important role. So 20% of the variance in DNA methylation is explained by genetics and also Agent X. So basically, what I'm trying to do in my work is to combine those genetics with DNA methylation and try to identify genetic factors that associate with DNA methylation at the CPG site. And where is that important? So if you're carrying, for example, an A allele, so you have this genetic variant, your transcription factor can bind, which makes sure that it gets hypomethylated and there will be no gene transcription. On the other way, if you're carrying of a G allele, so another genetic variant, then the transcription factor can't bind, get methylated, gene switch off. So so that's why it's important to look at that. So technically, how does it look like, or in practice? So your DNA methylation. So you have those microarrays that can measure 3 to 5% of the genome, not all 28 million. There are typically around 800,000. And basically, often we just measure methylation in blood cells. And obviously, your blood has a lot of cells. So in each cell, it measures whether something is methylated or unmethylated. And then you can get like a percentage of how much is methylated. So if you take like a measurement for one CPG, it can be either unmethylated with a 0, intermediate methylated is a half, or 1, it's completely methylated. So basically, if you take a blood sample, extract your DNA, measure methylation, you get a methylation level, which is between 0 and 1. And you can see that here in the table. So you have your CPGs here. So that's you have like 800,000 rows, and here you have each sample, and then you have all these different values. And in this particular CPG, this person is 90% of the cells from that sample is methylated. And what I try to do is try to identify genetic factors influencing methylation levels. So that was the DNA methylation set. On the other hand, we have on the same individuals, we have genetic data. So this is a genetic variant, and a genetic variant can either be you carry 0 alleles, 1 allele, or 2 alleles, and then you can correlate those. So here is your genetic variant, and there is a methylation value of 1 CpG with a linear regression model to see whether increase in the number of, in this example, A alleles is also associated with an increase of methylation. However, whole genome MQGL analysis, that one was just for 1 CpG, 1 SNP, is basically running a genome-wide study for each CpG. So you basically run 800,000 CpGs times 10 million SNPs. So 800,000 GWAS. So it's a massive amount of linear regression models. So it's really very computational. And each of those GWASs, so for each CpG, can identify a few hits. So you have an MQTL, for example, here, and also maybe here or here. So this is a huge undertaking. However, what we want, typically the cohorts that we have like typically DNA methylation and genetic data measured on around 1,000 samples. In Bristol, we have a bit more. But there are lots of other cohorts around the UK and the world that also have these data sets. So it's really important to combine those data sets, so you increase the sample size and you find more of those MQGLs, especially you have MQGLs that are really where the SNP or the variant and the CpG size is really close together. They have really strong effects, but then if they're a bit further away, they're more difficult to identify. So basically, and to do that across multiple studies, that's quite an undertaking. So often what happened before I started this project, people write a Word document with an analysis plan, and then they have to write their own script, or maybe get a script, and with often encourage errors made in the analysis. So what we have done, we generated a GitHub pipeline, where people can just download the scripts from GitHub and just basically run the scripts to calculate those MQGL. And it really improves the consistency, reduces error, it harmonizes the data. Methylation data is really prone to confounders, so you automatically can include like adjustment for confounders and patch effect. So it really improves reproducibility and power as well. And in addition, what I really liked about this format, it really enabled collaborations. I work with people I never would and never have met before. And also, if you have some kind of pipeline, also encourage like more new collaborations, so you can, with people who want to do different analysis, etc., etc. So basically, when I started this, so we brought all those cohorts with genetic and methylation data together in a consortium called the Genetics of DNA Methylation Consortium. So at the moment, there's more than 60 cohorts with genetic and epigenetic data on blood samples. And they're mainly European, so a few US cohorts and Australian cohorts, but the main focus is being European, and they typically have different types of arrays, but we're currently running analysis on the one with the epic arrays. So you really can increase your sample size from around 1,000 samples to like more than 60,000. And I come back on the multi-ancestry part later on. So basically, and what we did and what we released to them, so we generated a quite extensive GitHub repository. And basically what it does is people download those scripts, and then what they need to do, they prepare the genetic and methylation data, put it in their input data folder. Then they fill out a config file with the path where the scripts are, a path to the data, a path where the results going to be, and the process data. And basically, they there are several modules that kind of pre-process the genetic data and the quality control of the genetic data, pre-processing the methylation data. They kind of do adjustment of covariates. And then they kind of run GWASs of DNA methylations, but then equally you can also easily expand that to GWASs of other kind of phenotypes or clinical traits that you're interested in. And at the end, all the results that are aggregated summary statistics are generated in the results folder. And then people tend to upload the results, they encrypt the results and then upload it here to a collaboration space here in Bristol. And so far, we released the scripts in July, and 55 cohorts have now run the first module, which is like a data check. And then, so it's really going well. So quite importantly is like you can generate scripts, but if people don't know how to run it or what it's doing, that's not really helpful and also not really collaborative. So what we really have been working very hard on is generating a wiki. So first of all, explain what you're doing. That's not the only thing. But then also, there's like a frequently asked questions. There's like prerequisites. So how do you, what do you need to have? How do you prepare your data? What do we need to do to install the pipeline? And then when you have done all those steps, then you basically can run all the scripts from the pipeline, so the check data, process data, etc., etc. And this is an example of a module. So the first module is really pre-processing and standardizing the data, check if everything is fine. And then additional modules have been done that's really doing analysis. And basically every module has like a kind of module status and a person assigned to it. And then it says, oh, you can only start this module if you have run script 0, 1, and 2. And then there's also somewhere where, for this particular analysis, data is going to be uploaded to Google Drive. And then basically, there is like some instructions how to run it. So like now type 10a bash on your command line and execute that script. And because all the parts are in the conf file, it automatically does that. It's quite important when you do such an automated pipeline that you have kind of controls in there. So what we ask the developers is that they put in plots in the output that get uploaded so that you can check something. That it's generating a log file that checks whether the script actually has finished. There's all kinds of steps that we have built in and messages to make sure that everything works fine. And this is like an example of what gets uploaded to the server. For example, you can see a number of SNCs per chromosome or something, and you can see, is this person not missed a chromosome or something like that. One of the things that we asked was to generate an age distribution. And just as an example is then we combined all those age distributions in this plot. So here on the x-axis, each cohort that has run that analysis. On the y-axis is like the range, the range of ages of the individuals. So this is aggregated statistics, not individual statistics. And you can, for example, as an example, this cohort had made a mistake because there was like one person who was 120 years old. So we then go back to that person and say, your age is wrong. Is it in months? Is it what is going on? And can you change it? And they explained they had done some predicted ages, so there was a mistake, and then they had to rerun their analysis. Then the next step is what we do is like generate an MQGL catalog. So run all those 800,000 GWASs in each of those data sets. However, this is, like I said before, quite an undertaking, and there are several software packages that can deal with it. However, there's a problem there. So basically, those packages, they solve one problem, that is the computational. So they can run all those associations on a normal server. But the second problem is that we can't store them. they produce a huge amount of summary statistics in massive files. So there are two solutions there. So either you have to restrict to certain parts of the genome by taking some kind of biological hypothesis, or in our case, we found a different type of software, which is called HAZE, which was the only package that could store like a reasonable amount of summary statistics. But they don't do it like in the standard way with betas and standard errors, but they encode the data and using partial derivatives, which I'm not going into much detail on. And also I have to say that package is quite painful to use. So if anyone has experience with good Python programming, and because we want to extend that package, then I'm probably going to need to go to one of the options of the Gene Coding Institute to help with that. So we have used that. And basically, so the idea is then if we get like all the summary statistics back and we do like have all different types of MQTL using different types of statistics, we can then want to combine them in a big meta-analysis to build an MQTL resource for the community. And that is important because then people can download it, all your data, and then use it for their own GWAS, genome-wide association studies, epigenome-wide association studies, other kind of studies that they want to use it for. So we think it's really important that you make that open access. So everyone can download the complete data set, not only the significant ones, all everything. And what we're planning to do is more like, that's what our unit does, causal relationship. Is it methylation causing high LDL levels, or is LDL levels causing high different methylation patterns? That kind of stuff is what we do. Also with Dan Lawson, we work on natural selection, on functional properties, etc., etc. So there's a lot of things that you can do after you have built this resource. One thing before you release the data, and this is that you need to look at heterogeneity. So all those cohorts, they're from different countries. They have been collected in different ways. Different data sets, different arrays, different genetic data set, different methylation data sets. There's a whole range of factors that might be different between different cohorts, which might cause heterogeneity and bias the results. So what we tend to do is the heterogeneity analysis, and then in this case, basically you compare like kind of effect size across the different cohorts. And for example, this cohort had mismatched the genetic data with the methylation data, so they don't contribute. They just had an ID swap in one of the data sets. This was like a really good control check, and we moved that data set from our analysis. So that is my first part of the talk. So that was mainly about genetics of DNA methylation consortium. So that runs for quite a while, and like what has been discussed before, is like there's a big but there because it's only about Europeans. We really have an underrepresentation of non-European data sets. Well, that is one thing, because then you think, oh yeah, why do we care about that? And basically, why we do care about that is because the genetic variation is different in other populations than Europeans, but also the non-genetic variation, like the environmental variation, is different in other countries. And basically, if you can leverage that by combining those different population groups, then you get a whole load of new information about the genetic and environmental determinants of DNA methylation. And that's what I'm currently working on. So this is from one of my grants. And what we did there, basically, we want to try to combine genetic and DNA methylations data from across mainly the global south. So we have started a consortium with 20 partners from all around the world. So South America, we have people from Peru, from South Africa, Uganda, Japan, Indonesia, etc., etc. And then really we wanted to see whether those genetic factors of DNA methylation are different across different parts of the world. And that can also help with the integration of the already existing genetic factors that are shaped with disease. And one thing of that is, I have 2 slides. And one things of that is that we're trying to set up data shield. So data shield is a trusted environment, and where you can do non-disclosive federated analysis. So basically, what that means is you have like a central analysis service that we put in Bristol. Then you have data. You have all your data sets from your different partners. They stay at their institute on their servers, and then we can use data shield commands to extract aggregated summary statistics. And really, what we're trying to do is here, not like what we've done, like with the other approach, is really try to do like a pooled analysis. And one of the things that we're trying to do is try to identify the non-genetic variation of DNA methylation. And so this is a kind of principal component of plot. For each color is a different population group. And although this has been done on genetics, it has not been done on DNA methylation. And the only way to do that is in a pooled way. You have to have the data in one place, but we're going to do that with data shield and try to see why those population groups are different in their methylation levels. And the other way of doing is kind of doing an association analysis where your cases is one population and your controls is the other population, and see whether methylation factors are different across different population groups. And it's really interesting to do because some of those factors that might not vary between one population, if everyone smokes, you won't find anything because there's no variation. But like comparing different populations, you might identify new factors that you can't identify in a single population. So to wrap up, it's like I really like working like in an open science kind of way with GitHub pipelines that you make public, open access databases, open access publications. And also setting up an infrastructure that everyone can access, not only you in Bristol, because that enables collaborations, especially with people in the global side where they not necessarily have otherwise access to this kind of resources. Thank you for listening, and sorry for overrunning. Many thank you to the people in the IU, to all the people in the Genetics of DNA Methylation Consortium, and to the Diverse Epigenetic Epidemiology Partnership.